It's recording. And then, all right. Okay, so welcome to the 15th meme seminar. We have Viktoriia Kamska, who is a part of my cohort from 2014. She is originally from Ukraine, and that's where she did her bachelor's and the first master's in genetics and molecular biology in Odessa. And now she is based in the Max Planck Institute for Intelligent Systems in Stuttgart, where she's doing her PhD on the biomechanics of soft tissue of the lumbosacral region in birds. So the floor is yours. Kim, I think you're gonna have to let her share the screen. You have to make her co-host.
You're muted.
Oh, sorry. Yes, I'm just going ahead. You should be co-host now, so I guess you can present.
Okay, yeah, now I can.
All right.
Let me make the once. Okay, so yeah, hello everyone, but Charles already introduced me. And so my name is Viktoriia Kamska, and today I am presenting the 15th meme seminar. The topic is biomechanical interaction of locomotion and sensing apparatus in avians. And before to get to the announced topic, following the tradition of the seminar, despite the fact that Charles already briefly introduce my background, I would still go a bit deeper, and I would say like my bachelor and the first master degree I got at the Mechnikov National University of Odessa, and both these theses were focused on the phytotoxicity assessment of the technogenic waste material products before and after bacterial leaching. And after that, I was accepted by, sorry, I forgot about my animation. And after that, I was accepted as the master student of the Ludwig-Maximillian-University and the Uppsala University within the MIM Erasmus Mundus program. So at the Ludwig-Maximillian-University, I worked on my thesis, which was focused on the evolutionary morphology of the medium and small scale frogs. And at the Uppsala University, I worked on my master thesis at the paleohistological lab of the Professor Per Alberg. And my research was on studying long bone features of the extinct early tetrapod Hyneria. And this research project brought me the first author publication in the Earth and Environmental Science Transition of the Royal Society of Edinburgh. So that was the Fair Shift volume of honor of the retired Professor Jennifer Clark. And now, currently, I work on my doctoral research project at the Dynamic Locomotion Group at the Max Planck Institute for Intelligent Systems in Stuttgart, in Germany, under the supervision of Alexander Badrich-Prowitz. And I should say that this group focuses on underlying mechanism of dynamic legged locomotion in animals, and the group's research spans from bio-inspired and biomimicking robot locomotion, bio-inspired approach to sensor design, learning locomotion, and understanding locomotion biomechanics in animals and in robotic machines. And particularly my study focuses on bio and neuromechanics of a putative mechano-sensing organ in birds, which is called as the lumbosacral organ. And on this slide, you just can see the overall research project that are going to become the part of my doctoral thesis. And today I'll talk about already published project and introduce you into the currently running projects that has already been presented at some conferences. So let's start with my first already published project, which was published in the Integrative Organismal Biology Journal. And this project was focused on studying 3D anatomy of the lumbosacral canal in quill. And by working on this project, I aim to establish whether the topological and biomechanical features of more consistent with the fluid flow or the ligament strain-based mechanosensing function. But before I go deep into this research questions, first I have to introduce the problem definition of this research project. So birds live and hunt in almost every environment on Earth. They vary in size and mass proportion of body parts, and they exhibit high variability in their adaptation to the different locomotor ecologies. Birds' natural ability of agile and precise locomotion in aerial, terrestrial, aquatic, and arboreal habitat provide us a valuable opportunity to investigate the biomechanics and sensory motor control for bipedal locomotion. But then we have a question, like what distinguishes birds class among the rest of terrestrial vertebrates? It is well known fact that the vertebrates, they sense the linear and angular acceleration with the vestibular system of the inner ear. So the fluid flow, the circulating through passes through the semicircular canals and excites the sensors called the cupula. But what is interesting about birds, they are stabilized their head during walking and flying. Moreover, there is one interesting features that which mostly like distinguishing wandering aquatic, semi-aquatic, and terrestrial groups of birds. They have a long and flexible necks. And for example, there is the ostrich, and an adult male ostrich. The length of the neck of the adult male ostrich is about 1 meter length, and it's almost like the half of its entire body length. So with 1 meter of the leg length and 1 meter of the neck length, the information would take approximately 40 milliseconds to reach just only the brain, and so the even more time, the information passed through the nerve tissue, like even more time it would be traveled back to the spine. And just imagine that ostrich can achieve 40 milliseconds. It's really fast running. So in this case, we could expect the transmission delay. But as we know that the ostrich, they exhibit outstanding fast reaction speed. So based on this simple calculation, we can see that the vestibular system is not closely associated with the torso and limb sensor motor control for birds. And the absence of the sensory motor delay allows us to assume that there is existence of an additional mechanosensing organ that locates, where's my animation? Yeah. Which locates close to the central nervous system near the center mass of the synsacrum. And the avian lumbosacral region contains the several unique adaptations of anatomical structures that distinguish them from the rest of the vertebrates. So these structures include the glycogen body that is wedged between spinal cord hemispheres, the spinal cord, which is supported by dentata ligaments network, and accessory lobes, which potentially have like a mechanoreceptive capabilities that are organized pairwise in each segment along the spinal cord, and they are protruding bilaterally into the vertebral canal, and the remaining space is filled with the cerebral spinal fluid. And just a bit about the history. So the lumbosacral enlargement has been known over 100 years. And many researchers tried to establish a diverse range of the hypotheses about the function of the entire lumbosacral region and also about the function of just a single unique structures, for example, the glycogen body. Here is just an example of the most vivid hypothesis. So more than 100 years ago, Streeter is the scientist who suggested that the lumbosacral enlargement functions like a second brain in bird. And 20 years after that, Terny suggests, like he developed the hypothesis, just focuses on the glycogen body, that this structure is the nutrient storage. And next, vivid hypothesis on the lumbosacral region has been suggested at the beginning of the 21st century by Necker, who assumed that the lumbosacral enlargement works similarly to the inner ear. And he assumed that the accessory lobes, they accepted, like they function like this hair cells, which I introduced, called the cupola. However, Necker's hypothesis did not include any function for these unique structures like a glycogen body, dentata ligament topology, and our hypothesis, like we recently developed the hypothesis that allows us to assume that all soft tissue, not only just one part of the some structures, but in all the soft tissue inside of the lumbosacral canal enlargement, they interact in response to external acceleration, which is caused by bird's locomotion. And like essentially, we believe that the lumbosacral organ presents a spring mass system, which is damped by the surrounding spinal fluid. And we assume that the lumbosacral organ has the potential to act similarly to the accelerometer, and therefore external force caused by bird's locomotion leads the soft tissue to be deformed. And whereas the pre-stressed dentata ligaments network allows tuning the oscillation of the soft tissue, and because of the lumbosacral organ interacted directly into the spinal cord, it can transmit the sensor information much faster compared to other sensors we know. And coming back to the specific project, which is already finished. So it was focused on studying 3D anatomy of the lumbosacral spinal canal and quill. And for this purpose, I combined the digital and classical dissection. So for the digital part, I choose the specimen, like for the entire project, I use the specimen called the common quill, which is known as the Cattunix. And for the digital dissection, I processed my sample with the Lugol staining, and then it was scanned at the micro-CT machine at our collaborative group at the Royal Veterinary College in London. But for the classical dissection approach, I apply, of course, the same birds. But what the benefits of that approach was, the problem is about these fine structures, which I already introduced, called the dentata ligaments. And just to give you the reference, the dentata ligaments thickness varying between 50 to 100 microns. So with the resolution I got those time at with the micro-CT machine, it was almost like impossible. Like it was possible. I even did the reconstruction. You can see it here. But to be confident about the topology we observed, we had to repeat this studying this experiment and to conduct the classical dissection. And the results of the morphometric analysis of the space inside of the lumbosacral canal, it shows us that the soft tissue have the space for the potential movements of the soft tissue within the canal during the bird's locomotion, which has caused the oscillation of the soft tissue potentially. And the maximum horizontal and vertical, so we found that the maximum horizontal and the vertical expands, of course, at the glycogen body region. And we also extract the values, so approximately 15% of the cerebral spinal fluid inside of the canal is available for the up and downward movements. And for the side motion, it's approximately 16% of the space inside of the canal. And talking about the dentata ligament morphological features, which is really interesting because like we all have the dentata ligaments inside of our spine, but birds have like the uniqueness of birds' dentata ligaments network is about the topology, because here we have not only like the longitudinal, which we have, but also the middle one and the pairwise set of the transverse ligaments. And at the interaction point of the ligaments, they are spicing and they form so-called like nodes. So these nodes are also really interesting for our research, because they are very they have various degrees of mobility. So the longitudinal nodes are less mobile, because they directly attach to the vertebral canal here. But if we are looking for the middle one, so the nodes are like less limited by the attachment as for the longitudinal one, and that's why they possess like the higher options to be for the moving inside of the canal. So we assume that the topology of the dentata ligament adjustment adjusted to the glycogen body center of mass, because we conducted we didn't use any like a tensile measurement. We don't know the proper material parameters for the dentata ligaments. We had no access to this data, so we assume that we have like ideal version. So all our dentata ligaments, they possess like a uniquely distributed density, and we assume that the topology of like adjusted to the glycogen body center of mass, because we calculated the stretching of the transverse ligaments based on the space inside of the canal, and like based on this calculation, we found that the ligaments, which is close to the center of mass of the glycogen body, they are potentially less susceptible. So we can see it here to stretching, that the adjusted transverse ligaments oriented toward to the center of mass of the glycogen body at the obtuse angle. And now I would like to switch to the one of currently running project, which is the interdisciplinary project on biophysical model of the lumbosacral organ, which is already has been presented this year at the meeting of the Society for Integrative and Comparative Biology. And I work on this project in the collaboration with my colleague, doctoral student, Anmo. He is engineer, and postdoc of our group, Fernanda Brybieska. So on my previous project, like I already introduced you, I combined the digital and classical dissection approach to imaging and segmenting in 3D the details of the lumbosacral organ. But here it's important to emphasize that, the data, all the data which I extracted from the previous, like following the previous research, it was about the static configuration of the lumbosacral organ. But our task, like we want to test our hypothesis that the lumbosacral organ acts similarly to the mass spring-based accelerometer. That's why we need to investigate how lumbosacral organ behave during the body motion. However, lumbosacral organ deeply hidden inside of the multiple layers, like first it's the bone, which is the fused region. As we know, birds have multiple, and the lumbosacral region is the one of them. Also, it's covered with the multiple soft tissue layer, like the muscle, fat, leather, and that's why we found like there is no way currently available for us to conduct the imaging during the movements of bird, and to test our hypothesis on the behavior of the soft tissue inside of the canal in vivo. So instead of observing a potential soft tissue motion in vivo, we decided to develop the mechanical phantom, this one. So we hope that with the relatively truthful phantom design, we can create and characterize the soft tissue motion with like different morphological features. And in parallel, a mechanical setup allows us carefully control and measure the external acceleration acting on our phantom. So this project is like a pure interaction between the biology and the engineering. And mechanical phantom, it's really common approach to simulate the complex like real world physical effect. However, in our case, the lumbosacral organ include like its irregular shape, many tiny like features which was badly described in the literature. And that's why the implementing of this lumbosacral organ phantom, we call it phantom, is not a trivial task. On one side, we want to create the phantom relatively truthful to keep the most important features to understand how the LSO act in the real birds. On the other hand, we have no access to the actual biological tissue to work with. Which means we don't have the proper parameters for the tissue. We can't replicate the spinal cord softness. So we do kind of like a qualitative replication, and instead we need to recreate materials for the spinal cord, the glycogen body, the dentata ligaments. With like trying to achieve the good similarity. For our idea is to observe the behavior of the system, not to mimic it. And we approach our task by using already published results on study the coil lumbosacral spinal canal in 3D. And then we developed the biophysical phantom by simplifying morphological parameters of quill studied in 3D. And then we use the technologies for the soft robotics design to fabricate our biophysical phantom. And this is the step of the testing our hypothesis by using the oscillation simulator. So we already done this step. And this simulator, I should explain, it's produced the vertical oscillation, only vertical oscillation for the LSO phantom. And with the camera, here is the camera mounted, we already collected the data on the LSO tissue displacement. So we can observe like the camera is here, which means we look at our phantom from this perspective. And here you can see these black dots. It's the markers. And we're following the camera recording, and then we're using the special software to tracking the amplitude of the soft tissue inside of the tube. And that's how we're trying to analyze the displacement inside of the canal under the dynamic acceleration. So currently we're working on the data analysis. That's why I can't present you any results. And yeah, for this time, we're able to observe the soft tissue movements in our lumbosacral organ biophysical model. And our approach offers a blueprint to, like I already said, to the qualitatively study of the function of the lumbosacral region. It's not the mimicking. So we hope it will help us to understand how the lumbosacral organ behave in the real bird. And the last but not least project I will shortly talk today is the associating functional morphology of the lumbosacral organ and locomotion modalities in avian. This project also has already been presented at the this year annual meeting for the Society of Integrative and Comparative Biology. That's why I can talk about it. So the main idea of this project, as I already introduced at the beginning of the talk, birds, they are living in the different environments, and they're subjected to the selective pressure that modify their locomotion ability and the morphology. Oh, where's my mouse? Sorry. And the morphology also varies depending on the sensory need imposed by the locomotion conditions. So what we expect that the dentata ligament network morphology is related to the directional sensitivity and the frequency response characteristics. So the morphological variations of the endocast, you can see this is the endocast of the representative of the terrestrial locomotion, representative of aerial locomotion and aquatic locomotion. There is the features we are wanted to investigate separately on the endocast, and particularly I would like to pay attention on the so-called grooves, and their architecture. And we assume that these grooves could impact on the fluid flow inside of the canal. And here it's the path, how the fluid flow inside of the canal, and affecting the mechanosensing or like the accessory lobes, or magnitudes the soft tissue oscillation by enabling fluid flow around the moving soft tissue like a pump system. And yeah, to investigate if any dependency between locomotion type and the lumbosacral canal morphology, we decided to compare, like you already understand, landfowl, waterfowl, and passerine birds. And we choose the set of functional morphological landmarks based on the 3D segmentation of the lumbosacral canal endocast and related structures. So our first project was really helpful. It was kind of like the core, like the basics. And we are currently in the process of establishing the correlation between birds' locomotion modes and the functionally related morphological features we choose for this project. So to summarize the key message of both this running project, first I will give you a summary for the biophysical model for the lumbosacral organ, because this project is really close to be finalized. We're currently at the analysis step of our experiment. And what I can say that the extraordinary agility of avian locomotion could be enabled by the potential intraspinal mechanics sensing of the soft tissue motion, and like our approach with the developing of the biophysical model to quantitatively study the potential soft tissue motion. The next point, like we combine insight from the biology and engineer to tackle this biomechanical research question, which is the really great approach. I hope that like our approach to go all the way around will discover the feature which is currently hidden and unfortunately not available because of the absence of the good quality imaging facility. And yeah, like I said, we already conducted the experiment, and currently we are working on the data, and hope that soon you will see the publication. And talking about the associating functional morphology of the lumbosacral organ and locomotion modalities in avian. So this project mostly like at the initial stage. And here I would just give you that we aim to test if each locomotion modality develops its own finely tuned sensor adaptation. It's the core message, the core idea of this project. And for this purpose, we will work on establishing if the lumbosacral canal endocast and the related features, the grooves, if they varying depending on the acceleration imposed by locomotion ecologies. Also, we are aiming to reveal if the morphological variations correlate with the locomotion modes. And then, like I already previously said, we conduct a comparative analysis of the results between different groups of birds, the representative of different types of the locomotion. So this is the end, and thank you for your attention. I will stop sharing, or I can keep if you have a question. I would be happy to answer. And yeah.
Thank you very much. That was a great talk. I actually have a question, if I can go ahead. I really like your reconstructions and your images. And I was wondering what program you use for the reconstructions, for the rendering of your scans.
Oh, yeah, it's a great question. It's a methodological part of the question. Like in this case, I use mostly Hyneria. It's you know, there is Avisa
Yes.
and there is the Amira, so I'm using Amira.
Okay.
But also for the rendering and for the like morphometrics, because Amira, it's a bit clumsy in this case. It's hard to control. That's why I had to learn how to use the 3ds Max. So all this rendering and the most part of the rendering, not all, the part of the rendering you can see with the raw CT data. So this is the Amira. And if you see the structure or like the measurement I conducted, I used the 3ds Max.
Yeah, great. Yeah, I know it can take a lot of time. I did some work with Mira before, and it was, yeah. Sometimes it just takes ages until it uploads your data, and then you can't really do anything because otherwise it will take again 1 hour until it process one thing. But yeah,
and
it looks
sometimes
really
you
good.
need to put the extra filtering if the format of scanning allows you, because sometimes you get the pack with like the file format, which upload all the data, you can't see them, so they are not like a TIFF. And it's pretty annoying because you can't apply any filtering and you have to handle it somehow in the Mira.
Yes, big problem. Yeah, very interesting. Thanks.
Okay, any other questions? It seems like Charles really, really involved into his interview.
Yeah, I guess he probably still gone for a while, so, but we have the recording, so it's fine. I'm not sure if anyone else has a question. Please go ahead. Otherwise, we can wrap it up.
Hey, Victoria. I can ask, what are your plans after your PhD?
Hi, Frida. Nice to hear you. Yeah, I'm planning because I told you there is some project, like I showed this sketch and there is a plan of my research project, and I didn't include all of the like discussed and potential projects. So one of the like short time plan is to have some kind of like a short contract, maybe to work as the short-term postdoc, to wrap up all the projects which I can't just push into my PhD. And then I would like to looking for interdisciplinary project, also by doing it as the postdoc.
Nice. When are you planning to finish your PhD?
So my contract, like according to the Max Planck, now they changed the rules. In the meantime, it was possible to extend it like year after year. Now they have the strict full rules. And in my case, I already spent my 3 years, so I have just only half of the year. And yeah, within 3 months, I have to wrap up all the experiments and like manuscript writing, and then I have to use my 3 months for the thesis writing.
Wow, good luck with that.
Thank you. How about you? Are you already done with your PhD?
I'm finishing writing the thesis.
Oh, currently.
Yes, so hopefully we'll be defending in June, because it takes like quite a bit of time for people to read it, etc. Yeah, so wrapping it up.
And in total, how many years you spent?
Yeah, more than 3 years. It's gonna be almost 5 years.
5 years. Yeah. And in which country you're?
In Canada.
In Canada. I don't know if it's similar like in the US, because they have this master merged with the PhD, and for them 5 years it's just like most typical.
Yes, it's similar than the US, but here if you can either enter the PhD with or without master. If you enter with the master like I did, you have 4 years, but it can be extended.
Mm-hmm.
If you enter without the master, you have an extra year with funding, and then you have to get funding from other places, or I don't know. Yeah. So there's not really a limit time.
Maybe I need to stop sharing. question. Ah, it's here. Yeah. And how about you, Kim? You're already right now in the Max Planck Institute. Did you taste this? I don't know how it's interdisciplinary or not, because you're in the biological, you're in this B.Sc.
Yeah, I'm at it's the one for ornithology. I don't know if you know it, but it's only focused on birds, and it's definitely only biological. It's great, but due to COVID, it's, I think, not the usual atmosphere of Max Planck. At least I got told it's quite social, and they normally have a lot of talks and a lot of get-togethers, and currently, of course, it's really empty. So it's pretty much only a postdoc I'm working with and me at the institute every day. So I think it feels quite different, but it's a quiet environment. I like it, and yeah, I will see. I was supposed to be there for 10 weeks, and yeah, we had a lot of problems with the birds, and yeah, the chicks are not growing as fast as we thought. So I guess I will extend my project a little bit more and maybe just continue during the summer semester whilst I'm studying at LMU.
Yeah, it's a great issue that they allowed you to do this, to build a collaboration with the Max Planck, because when I come here in the engineering, for them it's really typical approach. They have like the student, like a heavy bachelor student, master student. So the engineers, they have to try them in the academy, and they also have to try into the industry. So they spend half and a half, and that's why the Max Planck Institute, they hiring the really talented student, and they do kind of like some part of the routine for the PhD or postdocs, which is like required extra hands, and for them it's really a great opportunity to learn and to sense this flavor, because we had some students which said like, no, I would like to go to the industry, I will not go to the research.
Yeah, that's true. I think it's a great opportunity, definitely, and you can learn a lot from interdisciplinary institutes.
Yeah,
I've never thought about that actually, but I guess you need also kind of, yeah, interest in physics or chemistry or something like that, so to actually understand what's going on. And I can imagine engineering is quite hard if you're not very familiar with this field. I don't know. Is it like, do you have a challenge sometimes with that?
Yeah, it was a like I don't know if you hear. I talked with Charles. It was a bit hard at the beginning to to find the approach how to set the experiment, because like biologists mostly, like what we would like to do, we would like to first open, like to do the dissection, observing that, and then to think what we are looking at and what we can expect for the next specimens, and we need a lot of the experiment. For the engineers, they design something, they control it, and then they record it with the high-speed camera, and then they try to analyze what they built. So it's quite different approaches, and biologists will require multiple, like the repetition steps. Engineers doesn't understand that, like if you plant this staining, the staining should work. Yeah, so, but I like it, and I have no regret that I decided to go into this kind of like a side direction from the biology, because it's really cool. It's like a pure interdisciplinary. That's what I really wanted. And it's possible to find some kind of new directions and application into the industry, because biologists, mostly like in the industry, is the genetics, not much for the morphologist.
That's true. So is your group mixed engineers and biologists?
No, it's the mixture of lots of people. Our group is really small, but yeah, the people, it's from very different environments, mostly of course from the engineers, but engineers also, like for them, we're all biologists, like molecular biologists and the morphologists. So we're all biologists. For them, it's not really clear what the difference is before you start to explaining. It's the same things with the engineers. For art, they're all engineers, but there is the computational engineers, which do the simulation, and they have no understanding how to handle the hardwares. And there is the people from the hardware side, and they do lots of this kind of like the drilling, skewing, and yeah, this like soft robotics fabrication. It's what I'm trying to learn and following them. It's really, really cool.
Very interesting, yeah. And about the organism, why birds? Is it like, do many people of your group work with birds, or was it more like because it just fits to your topic and to your interest?
Well, like our group focused on the different aspect of the locomotion, and because there is like a two main type of the robots. It's the bipedal, like a two-leg, and quadro pedal robot. So they try, like for the bipedal, of course, they're trying to get the inspiration from birds. And birds, like this specific region was selected because, like I said, 100 years ago, it was discovered, but nobody really understands what the function is. And like we build this hypothesis, and if this is the true, the contribution will be not only for the biologist, because it will fill the gap in the biological knowledges, what is the region is, what the function, because if you open the book, it's just like short description of the structure and no function, not really. They said like the glycogen body, maybe it's the nutrient and that's all, but there is no like a proofs. And for the robotics, it will be very, very helpful because they can use it for the exactly for the robotics. It will reduce the amount of the control, because currently this controlling issue caused lots of problem, and that's why the birds still clumsy. So we don't control each our step. We have multiple reflexes, and the birds, we can imagine, can stand on one leg and sleep in one leg. So it's that's why it leads to this idea. So maybe we need to look at birds. They already, like evolution already developed this sensing. So potentially it's hidden there, and if it's true, then it will be possible to replicate, and of course, but it's not my task. I'm mostly like the biological translator.
Yeah, in long term, but very exciting topic, definitely.
Yeah, challenge but exciting indeed.
Yeah, cool. Yeah, thanks a lot for that input. That was great.
Yeah, thank you.
I don't know if you want to wait for Charles. I don't know when he's coming back or if he's coming back, so I guess we don't have to.
We can talk 5 more minutes.
Yeah, I would say so. And otherwise, I think I'm also able to stop the recording and just send a link on to Charles or something, so he can do whatever he normally does with the link.
Yeah.
Cool. Yeah, really nice. So I don't know. Did you decide for the Max Planck Institute also because of the institute, or was it just because you were interested in the topic and it just fits?
I just was looking for, like, my goal when I was looking for the PhD position. Mostly I submitted to, like, when I check my email, it was the biomechanics. So it was my like wish, because when I was in Uppsala, it was the great group, and they gave me this opportunity to learn the paleontology, like the paleohistology. And I remember how my supervisor, she showed me this humerus bone. Like I worked with this piece of mineralized bone, which looked like a stone. And we used this synchrotron radiation facility to look inside. So we had 3 different resolution up to the osteocytes. And I could reconstruct the osteocytes. So those time I used B.Sc student Max. It's really, really powerful tool. And then like she showed me, like, see these features, these white features? It's the attachment of muscles. So you have not much time. It's just like a semester. Unfortunately, you have no time to develop your project into this direction. But if it would be super cool, and the PhD student just sit nearby me, he'd do this research. And then I thought, okay, that's what I want to do for my PhD. But I want not to study from biologist. I would like to go to the origin of the mechanics. So I start digging for the biomechanics from the mechanicist. And yeah, just because our supervisor, he's like not the trivial approaches, he decided to take the pure biologist, evolutionary biologist, to try.
Cool.
Yeah. So it was not like if to check in this institute, there is not kind of like there is for the molecular biologist. So maybe for some, I'm not sure if somebody in the MIM do the molecular biologist, but there is some molecular biologists, like a really strong group, they do the micro robotics. and they try to develop the robots for the treatment, for example, for the treatment of the cancer. So not to treat people with the radiation, or they try to find like a locus, and then this kind of like a car, or like a medical delivery. It's also like a cargo robotics. So, and for this purpose, they need the immunologist and they need microbiologist. So it's very, very interdisciplinary group.
Yeah, great.
It's like our group, it's the small group, but that is the department. But you already know in the Max Planck Institute, there is this kind of...
Yeah, exactly. Yeah, my group is super small as well. We are 4 people with me. And it's just getting smaller because one of the people is already almost finished with her PhD, and yeah, I don't know. I'm not sure if they want to have new people soon or not. I don't know. It's hard, I think, for the other PhD student in such a small group, because it's hard for her to contact people and to get help, and yeah, it's a little bit of an issue currently. That's why we also had to redo a lot of the methods, because she realized it won't work like she planned it, because she won't get any input from anyone, so she just has to do everything from scratch, which is interesting, and I guess it really teaches you a lot, but also it's hard.
Yeah, this is the research. This is the real research.
Yeah, definitely.
I hope you enjoyed this Max Planck environment.
I really like it. Yeah, as I said, I think it's different from what it is usually, but it's still nice. And I don't know how big your institute is, but the Max Planck Institute for Ornithology, it's quite small. So you kind of know the people walking around there, and it feels very familiar.
I see. No, the intelligence system, it's quite big, and it's young, but quite rich. So they have the great financial. So right now, the Stuttgart is in the Baden-Württemberg, and there is like 2 cities which form the Cyber Valley. And in this Cyber Valley, there is many institutes, like a Heritage Institutes, and the university, Stuttgart University, Tübingen University, and Amazon building right now. There's like a scientific city inside of these 2 cities, and our intelligence system have 2 buildings, one in Stuttgart, one in Tübingen. And the in Tübingen is the new building. Okay, Frida.
Yeah, bye.
Thank you. Bye.
Yeah, cool. Yeah, I've been to Stuttgart before. I think I've seen the buildings, but it's quite a while ago. But originally I'm from a place pretty close to Stuttgart, so that's where I was born. So I know the area a little bit.
Heidelberg?
Yeah, Heidelberg. Yeah, that's, the university there is like really high recommended university in Germany, and everyone wants to go to Heidelberg. I was accepted into the bachelor's from Heidelberg, but I decided that it doesn't fit me to be completely. So I went to Göttingen, if you know that. That's in the middle of Germany, a little bit further up. But yeah, Heidelberg is pretty, and Tübingen actually as well. Have you been there?
Yeah, of course I've been there, because I said intelligence system is the 2nd building, and before this COVID we have multiple talks there, and also I did part of my project in Tübingen.
Okay, yeah, great. Yeah, it was also on my list for the bachelor's, but yeah, again, so I decided for Göttingen.
Kleiner, aber fein.
Definitiv. Yeah, it's great.
Okay,
yeah, right.
So
thank you very much for that. I'll stop the recording now.